And so, magnifico rettore, magnificent rector, dear colleagues, it's a great honor and pleasure for me to introduce Professor Svante Pääbo, Nobel Prize for Medicine 2022, who will present the lecture titled A Neanderthal View of Modern Human Origins, which opens the 335 year of activity of Academy of Sciences of Bologna. Professor Pääbo was born in Stockholm, 1955. He's a biologist and a genetist. He's achieved the PhD in biology at the University of Uppsala in 1986. And since 1997, is the director of the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany. For his outstanding contribution, he is recognized as the founder of the paleogenetics. Before his studies and researches, in fact, the knowledge of human origin and evolution was only based on the analysis of morphological traits of fossil remains and of the lithic technology. But after the advent of genetics in biology, the scientific approach to bioanthropology was revolutionized. Professor Pääbo was a pioneer of the studies of biomolecular paleontology, starting in the 80s with the Egyptian mummies and subsequently in 1993 with the bones of Otzi. You remember the man of the glacier who was found in Val Venosta at the border with Austria. At the beginning of this century, he started the program to reconstruct the entire genetic heritage of the Neanderthal man. And in February 2009, at the annual meeting of the American Association for the Advancement of Science, he announced that the Max Planck Institute completed the first version of the genome of the Neanderthal. The conclusion of Pääbo was that we and the Neanderthal are two different species with a common ancestor many thousands of years ago. Even though in our genome, 1 to 2% of the Neanderthal DNA is contained. Some years later, he studied the genome of the Homo denisova, found in a cave of Altai Mountains in Siberia. He found that the mitochondrial DNA of the Denisovians was different from that of the Neanderthalians and from our. For all these discoveries in the fields of human evolution, he received the Nobel Prize of Medicine in 2022. But apart from the Nobel Prize, he received many other important awards, including the Leibniz Prize, which is the most important scientific recognition in Germany, and the Kirsteler Prize from the Foundation for the Future, and also the Theodor Bucher Medal. So thank you, dear Professor Pääbo, on behalf of all the members of the Academy of Science of Bologna, for having accepted the invitation to hold a lecture at this opening ceremony. I hope that you and your wife will keep a good memory of your visit to Bologna, which hosts, as you know, the oldest university in the Western world. Please, Professor Pääbo.
Started by thanking you very, very much for the invitation to come here. It's an amazing place. We arrived only yesterday evening, but we already realized that this is really a unique place. And we very much look forward to spending a few days here. So what I then wanted to do is give you an overview over what we work on with respect particularly to the Neanderthal and Denisovan genomes. Very good. Starting out by talking a little bit about the sort of technical efforts to retrieve genomes that are several tens or hundreds of thousands of years old, and then discuss what we can learn from those genomes about modern human origins and about what's unique with Neanderthals. So to start with, I then wanted to just remind you about what you all are aware of, I think, And that is that modern humans, that is the ancestors of everyone alive today on the planet, evolved in Africa and lived there around 300,000-400,000 years ago, so quite recently. And they'd lived there for quite a long time, accumulating genetic variation. And the amazing thing is that if you sequence genomes of present-day people, what you find is that there is more variation on average among Africans today than among people all over the rest of the world. Although, of course, many, many more people live outside Africa than inside Africa. And the reason for that is then that modern humans evolved here, accumulated genetic variation, and a part of that variation, so to say, went out and colonized the rest of the world. And with some genetic tricks, we can also figure out approximately when that was. And that's even more recent, of course. It's the big sort of exodus of Africa is less than 100,000 years ago, probably around 70, 80,000 years ago. But the interesting thing is then that when modern humans leave Africa, they were not alone on the planet. There were other forms of humans around. And most famously, of course, Neanderthals in Western Eurasia, and in Eastern Eurasia, other forms of humans that we just begin to learn a little bit more about. And our group are then sort of obsessed, if you like, with Neanderthals since 30 years or so. And we may ask sort of, why should we be so interested in Neanderthals? And to me, there are at least two main reasons for that. One reason is that they are the closest evolutionary relatives of everybody that are around today. So if we want to define ourselves as a group from a biological or genetic perspective, it's the Neanderthals that we should compare ourselves to see in what sense are we similar to them, in what sense are we different from them. And another reason is that they were here quite recently. They existed in this part of the world until about 40,000, 43,000 years ago. So say 1,200, 1,400 generations ago, there were Neanderthals here. So an interesting question is, how did we interact with each other? What happened when we met? And so on. But to then do that from a genetic perspective, we need to retrieve DNA from skeletal remains that are at least 40,000 years old. And that work then, as we heard, goes back to early 80s, when I started as a graduate student in Uppsala by studying ancient Egyptian mummies, which look very well preserved and are around 2,000 to 4,000 years old or so. So, for example, this mummy, 2,300 years old, we retrieved a small sample from here, looked at it in a microscope, and indeed in the skin here, you can see in the basal levels of the skin, things that clearly look like cell nuclei, where the DNA would be conserved. So we went on and extracted DNA from these such remains, and indeed found some human DNA in there, and published to great fanfare, saying you can retrieve genetic material that's at least a few thousand years old. The sad thing is only that over the next couple of years, it became quite clear for me and everyone else that those DNA sequences were clearly not from the mummy. Because there are lots of technical issues with this. They probably came from myself or from some museum keeper or from some chemicals I had used. Because it turns out that if you sort of compare the DNA you would extract from fresh tissue, from a fresh piece of bones, with the DNA you can extract from such old remains, there are a lot of differences. First of all, there's a lot more DNA, of course, in fresh tissue than in ancient tissues, often a millionfold more in modern tissues, or 100,000-fold more. The DNA is degraded to short little pieces. It has chemical modifications in it, and the vast majority of the DNA in such remains doesn't come from the mummy or the Neanderthal, but from bacteria and fungi that colonized the bones over tens of thousands of years. And a result of this is that tiny amounts of present-day human DNA that may come from a dust particle in the air or so wouldn't influence your experiments at all if you study present-day DNA, but may totally overwhelm the experiments when you work with these old remains. And that was clearly what had happened to me there. So over the next couple of years, we got more and more hysterical about cleanliness, treating all reagents you do to get rid of DNA, working under clean room conditions, and so on. And one worked not with human remains, but with extinct animals, such as mammoths, to work out the techniques, because it's then much easier to know that you have the right thing. If you work with a mammoth and find something elephant-like, but not identical to the elephant, you're very sure you have the right thing. If you work on an Egyptian mummy and find something human-like, it's very hard to know if it's a contamination or really an old DNA sequence. So we worked on all steps of this, how you extract the DNA from the bone, how you turn it into a form of the DNA, a library that you can sequence. What came around also was sort of the human genome sequence, so that we could make a data bank of the DNA in our old bones and compare it to the human genome. And we got a lot more efficient in these processes. And also the machines you use got a lot better. So somewhere in the beginning of the millennium, one could think about going to the Neanderthal and seeing if we could retrieve a complete genome or almost complete genome from them. The part of that was to find sites, archaeological sites, where conservation was particularly good in terms of molecular preservation of the bones. We focused in the end on one site in Croatia, the India Cave, and 3 bones initially from 3 different Neanderthal individuals. So we sequenced over 1 billion DNA molecules, taking into account the type of chemical errors, the chemical modifications that may cause errors in the sequences, and mapped these fragments to the human genome. And then by 2010, we had a first version of the Neanderthal genome, where we had a little over half of it, so we could begin to ask questions. And one of the first questions we were interested in was this question: what happened when modern humans came out of Africa and met Neanderthals? Did we mix with each other at all, or sort of did Neanderthals completely disappear? So an expectation, if we mixed with Neanderthals, would of course be that Europeans today should share genetic variants with Neanderthals, and they should do so much more so than people in Africa today. Since Neanderthals have never been in Africa, there's no reason to think that Africans today should have any special relationship with Neanderthals. But Neanderthals, of course, existed in Europe and Western Asia, so Europeans might have that. So we put together a consortium of population geneticists and started asking this question. We had the Neanderthal genome. We went out and sequenced 5 present-day people from different parts of the world, and we wanted to sequence them ourselves to have exactly the same error patterns, etc., in these genomes. So we had 1 European individual, 2 people from Africa, 1 from China, and 1 from Papua New Guinea. And then we started by doing a very simple test. We have sort of 2 present-day human individuals, and to test this idea, the 2 Africans here, we find all places where they differ from each other today. Then we have the Neanderthal genome, and we will see how often does a Neanderthal match this African versus that African. And that should be 50-50, right? Because there's no reason to think that one African is closer to Neanderthal than another African. And indeed, statistically speaking, that is the case. But if we then take a European individual and an African individual and do the same thing, To my surprise, I must say, at the time, you actually see more matching to the European individual than the African individual, suggesting that there could be a genetic contribution from Neanderthals to present-day Europeans. That surprised us, but what surprised us even more was that when we did this with a Chinese individual and an African individual, we again see more matching. Although most people would say there never been Neanderthals in China. And even more surprising, you look in Papua New Guinea, where everyone agrees there have never been Neanderthals, we again see more matching to the Papuan individual. So it turned out wherever we look outside sub-Saharan Africa, we see this excess matching. And the sort of idea that came out of that was to say that when modern humans leave Africa, they presumably pass by the Near East, where we know there were Neanderthals. And if these modern humans then mixed with Neanderthals and continued on and became the ancestors of everyone outside Africa, they could carry with them this contribution from Neanderthals. And the result is then that today, if your roots are outside sub-Saharan Africa, something between 1% and 2% of your genes, of your DNA, comes from Neanderthals. What has happened since is that we improved all these techniques, worked much more, so we come from this very bad genome we had in 2010, where we have fragments here covering it, but have big holes, to a situation where we have genomes where on average each site is covered 50 times or 30 to 50 times. So you have a much more complete view of the parts of the genome to which we can map these sort of short fragments. So you can now compare it much more in detail with present-day people. And if we do that in an example here for chromosome 9, and mark, or each line is one individual in Europe, and we indicate in red here parts of the chromosome that are identical or very similar to the Neanderthal genome, you will see that everybody carries some part of the Neanderthal genome. On average, that adds up to 1% to 2% of your DNA. But you also see that different people carry different parts of the Neanderthal genome. So you can also sort of jump from individual to individual and see how much can you puzzle together from the Neanderthal genome by looking at people in this room and a couple of thousand people. And that's indicated in blue down here. And that's in the order of 40, actually more 50, 55% of the Neanderthal genome is still running around on two legs today. So if you like, the Neanderthals are sort of not totally extinct. They sort of live on in present-day populations to some extent. Another thing that we found that early on was from this site in southern Siberia in the Altai Mountains, the Niseva Cave, where the archaeologists who worked there had found this tiny little bone. They were very skilled to realize that this might come from a human. It turns out to indeed come from a human. It's the last phalanx of the pinky of a girl. And it was well preserved, so we could reconstruct the genome of this too. And we were surprised to find that it was not a modern human, not a Neanderthal, but something else here that went back quite far back when we count the mutations in the genome to a common ancestor here shared with Neanderthals, somewhere in the order of 400,000 years ago or so. So we realized we had sort of stumbled across some form of now extinct human, distinct from Neanderthals, that we then, after lots of considerations, decided to call Denisovans after this Denisova cave, just like Neanderthals are called Neanderthals after the first place, the Neanderthal, where they were found. And we could start asking, have these Denisovans also contributed to present-day people? And indeed they have. So if you look in Western Eurasia, we find no contribution. If we look in Eastern Eurasia, we find a small contribution, less than 1% for sure, from these Denisovans. But interestingly, out in Oceania, in Papua New Guinea, Aboriginal Australians, 5-6% of the genomes come from these denisovans. And not only that, so if we sort of look, say, in Papua New Guinea and do comparison to the Neanderthal genome here and to the Denisovan genome there, we see fragments here that are quite similar to the Neanderthal genome that comes from Neanderthals. We see contribution from Denisovans that's actually more distinct from the Denisovan genome. One would be total identity. This is sort of less similar. But we see there's two contributions, Neanderthals, Denisovans. If we, on the other hand, look in Japan or in China, we find this Neanderthal contribution. We find this contribution much, much smaller, but it is there from Denisovans. That's similar to this one. But we, in addition, find a contribution from some Denisovans that are very close to the genome we have sequenced, but is distinct from this. So the conclusion is that at least two different distinct Denisovan populations have contributed to people in China and Japan. In Papua, it's just one of these who have contributed. But it also shows that whereas Neanderthals seem to have been rather homogeneous, these Denisovans had distinct populations. So they were probably much more numerous than Neanderthals actually, and had a much more interesting population history. So to just summarize this first part of the half of the talk then, there is an origin for Neanderthals and Denisovans, we believe, from studying the genomes in Africa somewhere. Their ancestors come out more than 500,000 years ago from Africa and evolve in Western Eurasia to what we call Neanderthals, and in Eastern Eurasia to what we call Denisovans. Where the border is between this probably shifted across time. We know that at this Denisova cave, there were Neanderthals at some times, Denisovans at other times, for example, and sometimes they even met. Then modern humans evolve in Africa. They spread across Africa and then come out of Africa somewhere after 100,000 years ago and probably mix early on with Neanderthals. Continue to spread and mix several times with Neanderthals actually, and in the east with several different Denisovan populations, and continue to spread out to other parts of the world where modern humans have never been. And these other forms of humans, then Neanderthals and Denisovans, disappear. You may ask then, are Africans today fundamentally different from other people in that they don't have this contribution from earlier forms of modern humans? Well, we don't know, I should say, but modern humans obviously evolved somewhere in Africa, spread across Africa, and since one outside Africa mixed with other forms, presumably one did so also in Africa. But we have no genomes yet from now extinct forms in Africa. But when we get them, I'm pretty sure we will find the same things going on in Africa. An interesting thing is that we begin to get a very direct view, actually, of this mixing with Neanderthals now. And the first case of that was a site in Romania called Oase Cave here, where cavers back in 2009 or 10 found this modern human mandible. Sort of morphologically looked like a modern human. It's dated to around 40,000 years ago, so it's a very early modern human in Europe that lived at a time when there may still have been Neanderthals around. So we were, of course, very interested if this individual had already mixed with Neanderthals or not. And this is Mattia Heidenjak's work, particularly on these late Neanderthals here. So when we got genome information here and look at each chromosome here and mark in blue things very similar to the Neanderthal genome, we were quite shocked to see that not only were the here and there were even huge parts here, sometimes even half of the chromosome or so that comes from Neanderthals. So that, of course, shows that this individual had a Neanderthal person in its family history quite recently. So from the size of these fragments, we can sort of show that if this is an individual here, because if you go back, you have 50% of your DNA from the mother and from the father, 25 on average from each grandparent and so on. And this individual then had a Neanderthal in its family history 6, 5, or 4 generations back. So we've almost caught someone in the act of mixing with the Neanderthals. Thought that that was a very unique situation. But in the meantime, there's also a site in Bulgaria, Bachukiro Cave, where one finds technology typical of very early modern humans. And one finds remains, teeth of modern humans that are about 45,000 years ago, deep in the layers, bit higher up 10,000 years later, other modern humans. And when we look at these genomes, we again find that these 3 individuals 45,000 years ago have close Neanderthal relatives in their family history. 10,000 years later, it looks like anybody around at the moment. So in the meantime, Johannes Krause's group have also studied in Leipzig, have also studied early modern humans, 40, 45,000 years old, further north in Europe. So those individuals are quite interesting, are quite close to each other, distinct from these in Bachukiro and Vase here. They live sort of further north than what one believes Neanderthals lived, whereas these individuals here lived where also Neanderthals existed, and they have mixed with Neanderthals recently. These have not. So it sort of seems to turn out that when the first modern humans who came to Europe overlapped with Neanderthals, they mixed extensively with Neanderthals. All 4 individuals with their genomes have individuals in their family tree. So it's sort of coming slowly to a view, I think, where one reason why Neanderthals and perhaps the Denisovans disappeared may not at all be that one killed them all or so. They may simply have been assimilated into modern human populations that came. But the sort of big question in the room is, in a sense, why did this happen? Why were Neanderthals assimilated into modern humans and not vice versa? Why were modern humans not assimilated into Neanderthals? And we discuss that a lot these days. These are sort of 4 ideas that one might think about that are not mutually exclusive, that one might consider. One is that there are several indications that modern human populations were bigger than Neanderthal populations. And one indication of that is genetic variation. If you look at sort of Africans today, they have quite a lot of genetic variation, as I said. People outside Africa have less genetic variation, but Neanderthals have even less variation than that, suggesting that their population size was small. And there's sort of archaeological evidence for that too, say in southwestern France. If you look at the number of sites for late Neanderthals and early modern humans, there are much fewer Neanderthal sites than early modern human sites. And these sites also seem to have been less intensely used in Neanderthals. There are less stone tools you find at the sites, less butchered bones, and so on, suggesting that modern humans were more numerous. And of course, on a grander perspective, modern Neanderthals were probably never more than 100, 200,000 people at any one time. Modern humans with time become millions and eventually billions of people and spread, not just staying in the old world, spreading to Australia, Americas, and so on. So there's a big difference in population size. Maybe that has consequences, or we don't know sort of what is a consequence of what. But it's also clear that modern humans had a much faster cultural evolution than Neanderthals. Neanderthal stone tools at the beginning of their history, 400,000 years ago, at the end of their history, 40,000 years ago, look quite similar. An expert can explain to me that there are some differences, but I need an expert to help me to show me that. Of course, modern human technology changes very rapidly. I need no expert to tell me that our technology 10,000 years ago was different from today. And it also, modern human technology becomes regionalized. An expert can immediately say if modern human stone tools come from Western Europe or from Central Asia, whereas Neanderthals were very homogeneous. This may have to do with population size. It may have to do with something else, of course. And one expression of this that one could think about as important may be, for example, projectile weapons. The oldest evidence of a bow, what is probably a bow and arrow, is around a bit over 60,000 years ago in Africa. But clearly when modern humans evolved. And then these microliths are probably sort of the points of arrows spread and are found in large amounts where you find modern humans. Finally, there are also more subtle things, perhaps. Art that depict things that we recognize what it is comes only with modern humans. There are some evidence of Neanderthal art, cave paintings that have been dated to a time before modern humans appeared, so they should be made by Neanderthals. That is quite a bit debated, but sometimes I kind of joke and say, if I look at what is Neanderthal art, it looks like very modern art to me, since I don't see what it sort of depicts. Whereas figurative art, that I immediately recognize what it is, comes to modern humans. So there are a number of differences there. That is probably linked to why modern humans become so numerous and spread. And if there is some sort of biological readiness for this sort of cultural evolution, we sort of dream about these days that we might be able to approach that by these genome sequences. Because when we now have good genome sequence from the Neanderthals here, and of course from thousands of humans and from our closest relatives, the apes, we can look for the genetic changes that are shared between Neanderthals and modern humans. They must have happened here. We can find things that are unique to the Neanderthals that happened there. And we can find things that are unique to modern humans and spread to all modern humans and happened here. So just now, for the last part here, I want to give you one example of what we study here and a few examples of what we study there. So if we begin with modern humans, we're so interested in these things that happen there because there may be functions unique to modern humans that hide or are caused by these genetic changes that happens here. So things that happen on the lineage of modern humans and spread to everyone today, or almost everyone. So we can list those things now by studying the genomes, and there are not that many. So there's around 30,000 changes in total, which is not that much. You can scroll through them in an afternoon on a computer. The problem is you don't really understand what you look at, because we are terribly bad at just inferring what a genetic change that we see causes in the organism. There are just around 100 amino acid changes, that is changes in proteins, so the molecules that really have functions in our cells. And we have this bias, of course, that we think that changes that may influence the function or the development of the brain are particularly interesting. So here are these sort of protein coding genes with these changes. We've studied several of them by now. I just want to give you the flavor of what we do by one example here, one gene called TKTL1 that we have studied together with Wieland Huttner's group in Dresden, and Anneline Pinson has been a postdoc involved in this. So this is an enzyme in metabolism, in energy metabolism, that then carries one change in the protein that is unique, that occurs in most people today. And what we are then doing is we're taking human stem cells and we change them back, their genome, to the ancestral state. So using this sort of technology that is now around, where you can go in and manipulate the genome in a cell. And these stem cells we can then differentiate into brain organoids. So these are little balls of cells in tissue culture that mimic the early steps in brain development. So we can then compare a modern human like TKTL1 making such brain organoids with an ancestral one there and look for differences. And you can then look in the epithelium in such things where brain cells develop, stem cells divide back here, they migrate up and become neurons in the brain here. So if we then do that and look at one part, type of these stem cells here, the basal radial glia, comparing the ancestralized to the modern human version, there seems to be less stem cells in the ancestralized version here. And then the cells migrate up here and become neurons, and this seems to have consequences in that there are less neurons made. So as you may know, Neanderthals have just as big brains as we have, but we don't know how the brain is organized. This could suggest that the proportion of neurons in the brain was less compared to other cells. This sounds very interesting. So, and a big question from this and from studying some of these other genes is did neurogenesis in modern humans differ from that of Neanderthals? But there are a number of caveats to consider here before we make too big conclusions. One is that what we study here is really early steps in brain development. And we know that that could be compensated later during development and may not influence how the adult brain functions. And of course, most traits, for example, how the brain functions is complex in the sense that many genes influence these traits. So it may be that any single such change doesn't have very big consequences. And in many cases, there are people around, and that's the case for TKTL1, that some people today carry at least one copy of this ancestral version and still are totally healthy and don't show up in the hospital with any problems. So I think we need to, if we want to figure out what makes a modern human modern relative to Neanderthals, it may be useful to compare to what morphologists do. When you find a skeleton and say it's a Neanderthal, you say it has certain features, big brow ridges, occipital bun, a barrel-shaped chest, etc. But for each of these things, there are some people around that have these traits just as prominent as Neanderthals did. You can find a person today that have brow ridges like a Neanderthal, but what you cannot find is someone who has a combination of all these things. So it's really the combination of these things that define us. And it may well be that this is a case for brain development or other things also. That we should think about this list of things that say that we are modern because we carry most of these changes in a derived modern human forms. But any single one of them is not necessary, of course, to be a modern human. But that's where we are in our work and thinking of this. We're, of course, particularly interested in, after all, finding some changes that are particularly important or have particularly big effects. So towards the end, I then wanted to spend some time on changes that happened here on the line to Neanderthal, that then define Neanderthals, and sometimes they come over to present-day people, giving you some examples of that. One of the first we studied was an ion channel. So this is a protein that sits in the cell membrane and lets ions through, sodium ions through. And Hugo Seeberg, who now has a research group in an institute in Leipzig, was at the Karolinska then, is particularly interested in ion channels. And he noticed that in all Neanderthal genomes we had, there were 3 changes in the protein here, which is unique. It's one of the very few proteins that have 3 changes in Neanderthal. And this ion channel is very interesting. It's not just any ion channel. It sits in the peripheral nerve endings and are involved in the initiation of pain. So when you hurt yourself, this ion channel opens, excites the neurons, and you register that as pain. So what we did was to express the Neanderthal version of this channel and the modern human version and test it in human cells. And you could see that for a certain stimulation here, the Neanderthal version lets through more current through the membrane than the modern one, as if it was more sensitive. I'm not a neurophysiologist, but you could then show that that's not because the channel opens faster, but once it has opened, it takes a longer time before it's inactivated and closes again. So it remains open longer for a certain stimulation. We could then make combinations of these three changes and show that it's two of them that's responsible for the full effect and one has no effect that we can see. So we thought that this was unique to Neanderthals. But another really interesting development in the field is that we begin to have huge data banks, biobanks in the world, where we have the genome sequences of people and medical information for them, and they've answered questionnaires. We can begin to associate genetic variants with disease susceptibility or other things in people's lives. And one of the most accessible of this is the UK Biobank in Britain. At the time, they had 360,000 individuals. And when we looked there, we found that some of these people, few, but some, did carry this Neanderthal version that had come over then thanks to this mixture with present-day people. So that then opens possibilities because we can then use modern humans as a sort of test animal and look at these people and see what effects does this Neanderthal variants have in people today. So there were 19 questions in the questionnaires these people had answered that had to do with pain. All kinds of pain. Do you have back pain, stomach aches, headaches, what have you? So if we sort of pool all these questions to sort of as a composite of how much pain you sort of experience in your life, we can say, what does that associate with? And since this was the first time for me to sort of have access to so big data, I sort of say, what's the biggest correlation with pain in your life? And it's very sad as you get older. Age is the biggest correlation. The older you get, the more pain you report in your life, which is sort of trivial. You have more medical problems as you get older. But more relevant then is that people who carry this Neanderthal version here do report more pain in their lives than other people. And if we relate that to this age effect, it is as if you carry this Neanderthal version, it is as if you were 8 or 9 years older in terms of how much pain you experience in your life. So this, of course, doesn't mean that we can say that Neanderthals did experience more pain, because we all know that how we experience pain is not just a question of the receptors in the nerve endings. It's modulated in the spinal cord and very much in the brain. Even the same stimulation can sometimes be pleasurable, sometimes painful in your life. But it's still interesting that all these people we have there, it's so rare, so they're all heterozygous. They had the Neanderthal version of just one of their chromosomes, the modern version on the others. Neanderthals were homozygous. They had this on both their chromosomes. So maybe we have to sort of modify our view of Neanderthals a bit and say they were not these insensitive, brutish people. Maybe they were actually oversensitive and everything hurt when modern humans came, so they become extinct. So another example of things that have sort of medical relevance today that come from Neanderthals involves a hormone that I think many of you have heard about, progesterone, which has many functions, but is produced particularly by the ovary after ovulation and prepares the uterus for a possible pregnancy. And we are now interested in the receptor for progesterone that sits in the target tissue. Progesterone binds to this receptor and elicits its effect. And there is a version of the progesterone receptor that has this distribution across the world. It's almost absent in Africa. It exists outside Africa. Whenever you see a genetic variant like this, it almost always comes from Neanderthals. And indeed, others had already noticed this comes from Neanderthals. And it's sort of was known even earlier than the Neanderthal genome that this variant is associated with increased risk of premature babies or preterm births, which is, of course, a risk for the baby, particularly in a society where you don't have medical care. So it had been speculated that this must be a selective disadvantage for Neanderthals and so on. Another cool development in the field is that we're now beginning to have very many ancient genomes of modern humans over the last 10, 15,000 years. So sort of many, many more genomes being sequenced, particularly from Europe and Western Asia. So we can sort of follow over time the frequency of such alleles over the last 10,000 years and see, do they go up? Do they go down? What happens to them? So if we do a little animation here and start 15,000 years ago and follow this Neanderthal progesterone receptor variants over time, then the red ones are just the ancient archaic genomes, black ones are carriers of this, and gray ones are non-carriers. And if we now go forward in time, around 7,000 years ago, we will see that this almost sort of explodes, which makes no sense. Why would something that increases the risk of having premature babies, put the babies at risk, increase? This was already 4 years ago when we didn't have so many genomes. Now we have over 20,000 genomes in this. We sort of see this drastic increase here between 9,000 and 7,000 years ago, and maybe a decrease here, but it's particularly this that is striking. So that made us go back to the UK Biobank again and then say, this is associated with among 400,000 people in Britain today. So this is unfortunately a bit backwards. Now we see what is a modern version, the non-Neanderthal version associated with. And then we find that the modern version is associated with increased risk of bleeding early in pregnancy, increased risk of miscarriages, and negatively associated with the number of full sisters you have, and actually also brothers, and the number of siblings. So the situation seems to be that this progesterone receptor, the Neanderthal version, it is indeed associated with increased risk of premature babies, but it's also protective against miscarriages earlier in pregnancy and actually results in more live births. So it seems to be sort of an evolutionary trade-off, that this Neanderthal version saves pregnancies that would otherwise be lost in a miscarriage, but the price you pay is that some of those babies are born early, but it's on average results in more live births. And we begin to understand why that is, because if we look at the Neanderthal progesterone receptor and look at its expression in the target tissues, if you carry this Neanderthal version, you express more of the progesterone receptor. So it's easy to imagine that if you make more receptor, you actually have more of a progesterone effect. And that's compatible with other studies in the meantime, where one has given progesterone to women that have experienced several miscarriages. And if you have more progesterone, you sort of increase the likelihood of successful pregnancy. So this is sort of the typical story almost of medically relevant variants that turn out to come from Neanderthals. Another small example are then 2 enzymes that are expressed in the liver and are very important for how you metabolize drugs you take. So and they clearly come from Neanderthals here. These variants of these enzymes are close to the Neanderthal genomes we have sequenced. So for example, if you take ibuprofen against pain and are a carrier of this Neanderthal version, you have a much better effect because you have a much longer half-life of ibuprofen in your blood. So you need to have better effect for the same dose you take. More seriously, if you take warfarin against blood clots, if you have an artificial heart valve or so, you need to decrease your dose of that to decrease the risk of bleeding complications, which could be serious. Finally, then, the biggest take-home lesson for me, how important the Neanderthal variants may be, came in the corona pandemic. Where, as you know very well, most people who got infected with the virus had no symptoms or very few symptoms. Some got very ill and some even died. We also know some of the risk factors, say male sex, old age, diabetes, what have you. But all these risk factors are not enough to explain why some people got so very ill and some hardly got any symptoms at all. So it was quite clear early in the pandemic that there must be genetic factors in the people too. So we had a small role in a big international consortium trying to look for genetic factors that influence severity of disease when you're infected with SARS-CoV-2. And in 2020, in the summer, the first results came in and they were unusually clear. So these are the different chromosomes, and you see that there's a risk factor, one big, huge risk factor on chromosome 3. And when we then looked at the DNA sequences in that region, we were very surprised because these risk variants here that infer risk, they are very close to the Neanderthals, particularly to the Croatian Neanderthal cell sequence there. Whereas all these other ones that are protective, if you like, are then modern human. So this risk variants clearly have come over from Neanderthals to present-day people and exist in some of us today here. So early in the pandemic in Scandinavia, if you were hospitalized with severe COVID and were not a carrier of this Neanderthal version, you had about a 7% risk of dying. If you were a carrier of the Neanderthal version, you had about twice as higher risk to die. So it is, for being a genetic risk factor, quite a strong one. So of course, there's a lot of interest in what is in this region of chromosome 3. Unfortunately, I should say, it's a very complicated region. There are several genes there. There are at least 3 genes where sort of variants have been suggested to be responsible for this, but we don't know yet. But we and others work on this. If we understood why this confers bigger risk of becoming severely ill, we could perhaps treat severe COVID better. If we look at how this is distributed around the world, it has this typical pattern of being not present in Africa, being present outside Africa. So we can sort of use this frequency to estimate how many extra deaths did we have in the pandemic so far due to this Neanderthal contribution. And that's quite shocking. It's at least 1 million extra deaths due to this contribution from Neanderthals. You can notice something else interesting here too, and that is that if you look in East Asia, it's almost absent in China and Japan. It's very frequent in South Asia. So we don't know why this is the case. It had clearly been selection in the past, sometimes against this in East Asia, and it's probably been positive to carry it in South Asia. You can speculate about other infectious diseases, say maybe epidemics of other coronaviruses in East Asia or so, but we don't know why that is. But we are beginning to learn that this region in our genome does influence susceptibility to other diseases. So Hugo Seberg again noticed that if you look at this region on chromosome 3 and go 1 million base pairs down, you find another gene that is quite famous, CCR5, which some of you may know as a co-receptor for HIV, so for the virus that gives you AIDS. So people who lack this protein totally on both chromosomes are actually protected against infection by HIV. So if you now look at the people that carry the Neanderthal COVID risk variant here, they express less of CCR5 than other people. And indeed, if you go in cohorts and look both in Finland and in the UK and in the US, if you're a carrier of the COVID risk locus, you have a reduced risk to get infected by HIV if you're exposed to the virus, because you have less of this protein on your cell surface. So this is sort of this locus and the variant that come from Neanderthals. It increases your risk of becoming severely ill if you're infected with SARS-CoV-2. But if you're exposed to HIV, it reduces your risk of being infected. So it's clearly a sort of double-edged sword. It's typical of genetic variants, I think, that is really depending on the environment, what effects they have. In this case, on what virus you're exposed to. And in addition, there's then been in East Asia and South Asia, factors in the past influences things. It's not just a double-edged sword, I would say, it's a sort of multi-edged sword. So with that, then I should come to the end and say, I hope that I sort of convinced you that it can be useful to have the genome of our closest evolutionary relatives, because we can then find things that are unique to modern humans here and try to understand those variants. We can find things that are unique to Neanderthals that have come over to us today and have consequences today. And the way we study that is then by these big biobanks, which are increasingly important to understand this and other things in biomedicine. We also edit stem cells and make organoids and also edit the genomes, although I didn't talk about that, of model animals such as mice and humanized and Neanderthal-like mice. And this is done mostly in Leipzig, in our lab there in Germany, this group, and a little bit, some of the work also in our small group in Okinawa, at the Okinawa Institute of Science and Technology in Japan. And with that, I then thank you for your attention.
Thank you very much, Professor PäÄBO, for your outstanding lecture.
No.
No question
No.
are admitted. Okay.